Welcome to the Emergency Metaversal Emergence Recording Interface Terminal Undersampling System. Today we have Item Hashtag SCP-047 Object Class Kether Special Containment Procedures SCP-047 is to be contained in a 0.5 mx 0.5 mx 1 m hermetically sealed storage box at all times. This box is to be locked in Storage Locker 047A, inside P3 Secure Biohazard Lab 047B. Any entrance to an activity inside 047B will be recorded by biometric scan, closed circuit camera, and redacted. Entry to 047B requires the authorization of the project manager, in addition to at least 105 level clearance. SCP-047 is to be treated as a priority for contagious biohazard in all protocols, including mandatory quarantine if exposed. Suite Q-047 has been provided, adjunct to Lab 047B, for this purpose. In the event of outside contamination of SCP-047-1, Lockdown Protocol 047-01 Yersinia must be engaged. Description: SCP-047 is a heavily rusted, breached gas cylinder made of an iron-redacted alloy. When exposed to open air, the material of the cylinder evaporates slowly, producing a previously undocumented mutagenic gas. This gas has no effect on eukaryotic organisms, e.g. humans, but profoundly alters prokaryotes, showing preference for common human microbiota, the natural microorganisms that live on the skin and throughout the body. On rare occasions these mutations produce a superbug, collectively known as SCP-047-1, a natural commensal with enhanced survivability and therefore opportunistic pathogenicity. The pattern of changes induced by SCP-047 suggests that these highly infectious microbes are, at least to some degree, selected for. Although the specifics of SCP-047-1 species are dependent on the base bacterium from which it is derived, there are several characteristics which appear to be generally consistent across all cases of SCP-047-1 mutation. Enhanced survivability in the bacterium's natural environment and similar environments. Full-spectrum antibiotic resistance. Increased reproduction rate and consumption of available material. Development of a sporulation ability in gram-positive bacteria. Increased ability to uptake, hold, and share plasmids, particularly in gram-negative bacteria. Increased transmission, due to traits described above. SCP-047-1 samples are normally debilitating and virulent. However, compared to other Kether-class SCPs, it should be noted that SCP-047-1 have a relatively low mortality rate due to their action through mundane biological pathways. Several strains of bacteria have been selectively mutated by SCP-047. Mutation of bacteria in culture is possible, but the process appears to be much more effective with bacteria living on a human host. Generally, mutation of natural commensals for experimental purposes is encouraged. After the containment breach of 30/01/2010, see Incident Report Yersinia 047-01, 2010. Incident Report Yersinia 047-01, 2010. SCP involved, SCP-047. Description: on 30/01/2010 at approximately 0300 hours, Storage Locker 047C, Site Redacted Lab 047B, containing bacterial samples mutated by SCP-047, was compromised after a complete simultaneous data expunged, leading to failure of security measures in the area. 3. Samples, of a total 12, were stolen. Data expunged since the initial containment break. Outbreaks of one of the stolen bacterial strains, Propionibacterium 047A, were recorded globally in communities of increasing size and population density. Further information on stolen material, spread, and containment follows. Compromised items, Propionibacterium 047A, Streptococcus 047C, and Clostridium 047A. See SCP-047 for further information. Outbreak information. First outbreak, P047A, 27/02/2010, Siberia. Contained. See Incident Report P047A03-2010. Second outbreak, P047A, 30/03/2010, Northwest Territories, Canada. Contained. See Incident Report P047A04-2010. Third outbreak, P047A, 29/04/2010, South Australia. Contained. See Incident Report P047A05-2010. Fourth outbreak, P047A, 27/05/2010, Mato Grosso, Brazil. Believed contained. See Incident Report P047A06-2010. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P047A and be on the lookout for possible infection. Fifth outbreak, P047A, 26/06/2010, redacted, Iraq. Site immediately data expunged which is believed to have contained the infection. Access to incident report denied without O5 clearance. Sixth outbreak, P047A, 26/07/2010, Cameroon. Quarantine enacted. Efforts to track outgoing civilians underway. Infection not contained. See Incident Report P047A07-2010. Seventh outbreak, P047A, 24/08/2010, Dalarna, Sweden. Quarantine enacted. Believed contained. See Incident Report P047A08-2010. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P047A and be on the lookout for possible infection. Eighth outbreak, not recorded. Believed to have taken place in North Korea. Data expunged. Agents with government access are attempting to gain access to parallel information, but due to data expunged, local services have been extremely uncooperative. Containment status unknown. Ninth outbreak, P047A, 23/10/2010, South Carolina, United States of America. Quarantine enacted. Efforts to track outgoing civilians primarily successful. One civilian in a pickup truck is believed to have data expunged. Infection not contained. Resolution. Reports from data expunged indicate no further outbreaks are believed likely, but agents are advised to be on the lookout for new flare-ups resulting from uncontained civilians in previous outbreak regions. These may continue for years to come due to P047 sporulation. Investigation into the cause of the initial compromise is underway. Anyone with useful information may anonymously contact security via the attached form. Mutation of already pathogenic species is banned and all existing samples must be destroyed. 3 particular species of SCP-047-1 mutated bacteria are of note, due to their involvement redacted/redacted/2001 redacted. Propionibacterium 047A is a strain of Propionibacterium acnes mutated by SCP-047. Pathogenicity: severe skin colonization around sebaceous glands. Modification of skin pH to levels that become toxic to skin cells. Massive inflammation and immune cell infiltration. Eventual breakdown of skin structure leading to sepsis. Transmission: transmitted by skin-to-skin contact. Can remain active on inorganic surfaces for up to 5 hours. Lethality: approximately 40% mortality rate. Runs its course in 2 to 6 weeks. Very visible symptoms within 5 to 10 hours, contagious within 2 to 5 hours. Handling: as soon as visible symptoms form, victims must be quarantined. Deceased victims should be incinerated. Streptococcus 047C is a strain of Streptococcus mitis mutated by SCP-047. Pathogenicity: causes inflammation of the mouth and esophagus initially. Leads to open sores in the mouth, which result in S047C entering the bloodstream and becoming septic. Death is usually due to infectious endocarditis. Transmission: droplet. Can remain active indefinitely by sporulation. Lethality: approximately 35% mortality rate. May become a recurring chronic condition if non-lethal. Handling: subjects with any sign of mouth infection should be quarantined. Deceased victims should be incinerated. Clostridium 047A is a strain of Clostridium difficile mutated by SCP-047. Pathogenicity: unknown. C047A was developed from tissue culture and has never been exposed to a human. No samples remain in Foundation control. Transmission: unknown. Presumably transmitted through fecal contamination, as with C. difficile. Due to smaller, more robust spores, may also aerosolise with flatus. Effects of aerosol intake of C047A cannot be predicted. Lethality: unknown. Presumed extremely high risk of destruction of endothelial lining of gastrointestinal tract, leading to inflammation, sepsis, toxic megacolon. Handling: until further research has been done, victims should be quarantined and placed under 24-hour medical observation to develop functional diagnostics for this strain. Deceased victims should not be incinerated until adequate etiological research has been performed. Recovery Log 047, SCP-047 was recovered from the Site Secure Laboratory by a Foundation Biohazard Recovery Team in response to a full compromise situation on redacted/redacted/1990 redacted. Testing logs indicate that the research team was attempting to contain data expunged in a class redacted SCP stable pressure cylinder, which led to redacted combining with redacted. A full molecular biological analysis of this is available in redacted. The initial release of gas when SCP-047 was structurally compromised was sufficient to cause a microbiota bloom of uncounted species of SCP-047-1, killing all staff in the lab within redacted hours. Exposed site redacted staff obeyed standard Foundation quarantine/containment protocol, and the infection was contained successfully.